Hello everyone, I'm Amun and I will present our work on management of pathology annotations for high-dimensional metabolomic data, particularly the application of DESI mass spec imaging for prostate cancer characterization. Histopathologic examination of tissues serves as the gold standard in cancer research. Digital annotations are labels for high-dimensional imaging data. But extracting and mapping these labels remains challenging. Traditional image processing applied to the snapshot of tissue annotation overlay limits the number of differentiable classes, which is particularly needed for heterogeneous tissues like prostate. It also ignores extra comments added during annotation, and the spatial resolution is limited to snapshot region. Here, we propose a workflow for handling histopathologic annotations, which is compatible with the standard platforms and addresses these challenges. The proposed method includes label extraction, tissue localization, dataset generation, and validation through deep model. The method is tested for prostate cancer grading using DESI mass spec imaging, which measure thousands of low molecular weight chemicals at 50 micron resolution. Our dataset consists of 38 needle biopsy cores from 19 patients, sample from radical prostatectomies, and cryosection into microscopic slides. A detailed multi-grade, multi-scale annotation protocol consisting of 9 labels was proposed based on the spatial distribution of cells. Particularly, the mixed label are proposed when low and high-grade cancers are evenly distributed and not separable in high power. Next, the annotation data, which contains boundaries, coordinates, and comments, were exported from the digital platform. Each got post-processed and compiled into a multi-layer label image. The metabolomic profiles of the biopsy cores were then collected and analyzed by DESI. Each pixel spectrum is reduced to its principal component to extract tissue latent representation. The label images were then spatially registered to these DESI tissue images, and the transformation was used for final dataset generation. The dataset was then used for prostate cancer grading in a deep architecture consisting of convolutional and residual layers. When used for supervised training in a pixel-based manner, high test accuracy of 82% was achieved. Also, using a slide-based analysis of principal components of DESI spectra, a high spatial correlation was observed between the representative ion images and the corresponding histopathology label images. Here we present 3 cases. These results prove the validity of proposed workflow. In conclusion, the proposed annotation and dataset generation is compatible with AI-oriented cancer research. The application is also extendable to other modalities and tissues. Thanks for listening.